Thank you. I don't even know, man. So the title's very grandiose. The abstract was over the top. I'm trying to lower the bar, right? There's a lot of this is really just a lot of my thoughts, where I'm going, what I'm trying to do. There's not a lot of, hey, look at this amazing thing that belongs in whatever journal, right? And there are some pretty pictures, but oh yeah, another reef talk. That's great. My perspective on reefs is not corals, it's the system. That's really what I try to come at it. Not that there's anything wrong with corals, I like corals, but I think there's a lot more going on, and I hope to sort of convey some of that. So the boilerplate that you're all aware with, my clicker isn't working. Click. Okay. Well, I'll just do it the old-fashioned way.
No, it's working.
Okay. That's what happened.
Yeah, I clicked out of it.
You clicked out? You what are you doing?
This away.
You moved it right in the way.
Sabotaging your eye.
Minimize. Oh, it doesn't minimize. Sorry, folks.
Tried that first.
How does it doesn't even move? Oh, there we go. Okay.
Yeah.
There we go. Hey. Boilerplate. Why are reefs important? Why do we care? And so when you go to NASA and they say, why should we give you this money and all that kind of thing, you have your reasons. Everyone's got their reasons, but why do people in Kansas care? And so you got to have all these things. And these are all the things that we sort of tell. And it's in every proposal that you write about coral reefs. Coral reefs are super important for all these things. I'm not going to belabor them. But they're important. Ecosystem goods and services they provide to us. And then we got to sell it, right? They're important for these reasons. We get this from them, and it's great. And that's still not working. Okay, I'm not wandering around. So what underpins those goods and services? And there's three pictures here. I was going to have it, whatever. But here's a pretty picture of a reef. Isn't that great? You want to go snorkeling there. But what underpins the ecosystem is structure and function. And ecology 101. Here's the structure, and you've got everything sort of highlighted and outlined and everything. There's the pieces there, and the pieces are doing different things, and there's different kinds of pieces. And and there's three-dimensional structure too. It's not just the biological things that are there, but their 3D structure. And then there's the function. And I have for function import, incorporation, conversion, turnover, excretion, export of materials, things that are going on. There's things are being exchanged, things are being eaten, things are being taken up and excreted. And it's all going on in the structure, and they go together, right? So we're really, really good in reef science at the structure part, like exceedingly good. We know how to put corals in tanks and do things to them and measure them and put them here. This says +4 degrees C. If you ever studied corals, you know what that means. Let's bleach them is what we're trying to do here. We're good at reef surveys. And this is actually from a summer class that I teach. This is a student. So there's a little plug. And surveying reefs and doing all kinds of things. We're really good at taking the tissue samples and grinding them up, or putting in liquid nitrogen and extracting metabolites and looking at the DNA. We're good at all. We're great at the structure part. Really, really great at it. And we've learned just an enormous amount in the latter half of my career, which I don't want to think about how long it's been. So, but the function part, I'm less happy with personally. I think we could do a lot more about reef function. So, for example, fluxes. This is the marine version of the flux tower, right? I don't know if you ever know what a flux tower is. We're all marine scientists. Flux tower, they put these towers over rainforests, and it measures oxygen and carbon dioxide going in and out. Here we are on a reef, right? And measuring flux. In this case of oxygen, but you can do whatever. Not a lot of people do this, probably because it's expensive and it's a pain in the butt. But it's important to understand fluxes and rates of things. How fast is oxygen being produced or taken up? And And what about nutrient uptake and excretion? Just want to throw this out there. Nutrients are not tightly recycled on reefs. Within organisms, maybe, but not within the system. Just throwing that out there. I can tell you about it later if you want. I'm not going to belabor it. But that's a common misconception. What about primary? Here's primary production. This is actually the same reef, just a map of primary production. It's not the same everywhere. That's what the different colors mean, right? And of course, you have circulation and connectivity and everything that goes with it. There's all sorts of other functions that are in there that we don't actually get a lot of in reef science. And I would like to see a lot more. I've put scaling up there. And scaling is because we can measure, I can go in the reef and measure productivity with this. But that's a 10 meter by 10 meter area. And that's a community. It's not a reef. We need to scale it somehow. We need to get from point measurements to ecosystem measurements. So, where do reefs live? They're in the red, right? The 9,000 reef systems in the world, of course, not exactly 9,000, about 500,000 square kilometers of total area. That's not a lot of area, to be honest, but it's spread over 200 million square kilometers of ocean. So the reefs are far and few between. Where they are, where they exist, is constrained by available antecedent topography. There has to be shallow water. If there's not shallow water, ain't going to be a reef there. And environmental drivers. That's why all the reefs are between about roughly 32 north and 32 south, because that's where the environment is good for reefs. Now, this is reefs, not corals. Talking about the systems, not the organisms. But organisms live there, and processes happen at different scales. So I want to point out, so there's a lot of intro stuff. This is like lecture stuff to you. I'm sorry for this. Go to sleep, take a nap. Mass transfer drives these processes. So mass transfer is the movement of materials or heat, even things from the water to the benthos, in and out, right? Okay. This is my favorite and least favorite axis label of all time. U is water flow. CF is friction factor. RE is the Reynolds number. SC is the Schmidt number. And S is the mass transfer rate in meters per second. And if you take whatever gypsum or phosphate or nitrate and you plug in the right values for the Schmidt number, I believe it is, I don't know, you normalize it appropriately, you get the mass transfer rate is linearly proportional to the water flow rate. And bottom friction is in there, sure, but biology isn't, which is kind of fun, right? This is all physical. Now we can get the flux of a nutrient, for example, by this. You take S, multiply it by the difference in concentrations. And that gives you the flux into or out of the seafloor. But the point is, the faster the water flows, the faster the uptake rate, transfer rate. I mean, it stands to reason, right? So I'll just read: organisms and communities that experience faster water flow can get nutrients and radiate heat more quickly, right? Faster water, they can do a little bit better. That leads to patterns and organization of reef development. The physics does. We'll get there in a second. This is just a little experiment that I did with an intern last summer in a little flume, corals in the flume, and measuring oxygen, doing flow respirometry. Flow speed, gross primary production. Okay, so yeah, the same community, right? Faster the speed, the faster the production. Community wasn't changing. We weren't messing with it. We weren't tinkering with it. It was randomized flow speeds, all that stuff. So that means that if a community, and here it's corals, it could be another community, live in faster water flow, they're going to be faster production. And that means more potentially more energy for growth and reproduction and things like that. So here's Anahuac Atoll. They blew a big bomb up there. There's the windward bit. There's the leeward bit. Windward gets all the trade wind swell, continual waves breaking, water flow, all that stuff. This is what the reef looks like under the high activity area. Leeward, in the lee, does not get quite so much. This is what it looks like. And it's not a coincidence that the reefs, and sure, it's beautiful, don't get me wrong, but I'm sure it's not a coincidence that the reefs look different. This reef is buttressing against the waves, and it's able to do that because it's able to grow because of the mass transfer, and it's feedbacks. More lecturing. So this is actually from a class after Darwin figured out how atolls form, right? Because we had that in an earlier lecture. The next reef problem was, how do they have such high productivity and biomass? Because here's this ring of an atoll in this deep blue ocean. Reef gross primary production is 25 to 50 times that of the open ocean, right? It's a lot. Really high rates, and the biomass is through the roof, and they do that in an oceanic desert, ridiculously low nutrients, really low. So how do they do that? Right, that was the question, and a lot of reef scientists still have that question today. They ask it, but as I said, we've just talked about it. It's mass transfer, right? And this is from Kinsey in '83. He said, look, reef areas, reef communities in high activity zones have higher rates, and it's because of that, which came later. So I won't go into all this reading stuff, but basically what happens is you got a parcel of water. The water doesn't have a lot of nutrients in it. If you're a phytoplankton, you're out of luck, right? But if you're a reef, you sit there all day, every day with water, 24/7, 365, no holidays. Water's flowing over you, and all those little parcels of water bring little bits of nutrient, but there's an infinite supply. So you effectively get all the nutrients you need, right? That's how it works. And I had this dig. It took a while to figure this out. Even today, meteor scientists don't appreciate the water flow. Okay, fine. Let's get out of water flow. Let's talk about light. So if you've been snorkeling, you've been diving, you've been on a reef, you've been around Tampa Bay, you know the water's not the same color everywhere, different clarity, right? So this is for reefs, and this is showing the spectral irradiance of downwelling light as a function of wavelength at different depths offshore. And you can see that blue light, there's still 50% of blue light surface at 20 meters. Red light goes away fast, right? On the fore reef, now you're coming in from the ocean, but you're still outside the reef. It actually the reef does influence that water. It gets a little bit less clear. So the blue light disappears a little bit faster, and actually you're getting a little bit more of what's green, the green peak here at 20 meters depth. On the reef flat, it's even more turbid. And so if you went down to 20 meters, there would only be 10% of light, except reef flats only go, sorry, anyway, reef flats only go down to about 2 meters. So that's not a problem on reef flats. But in the lagoon, it can be quite, these are average values, by the way. By 20 meters, you've only got 5% of the surface light. So where you are on the reef sort of matters in terms of how much light you get. Here's a nice picture of a former technician measuring light, or setting up the instrument to measure light. So biologically, here's a minute of photosynthetically active radiation at about 3 meters depth, maybe 4. 1 minute from one day, from 12:30 p.m. to 12:31, and over that 1 minute, light varies by a factor of 2. So you're an organism living there. That's what you're dealing with, right? And it goes up and down pretty quick. And on longer time scales, here's 5 days. This day was a cloudy day. This day was mostly cloudy, but patches of sun, and then cloudy. In organisms, you're dealing with this. The community, the reef, is dealing with this variability in light that happens from milliseconds to years because of the seasons. Light is constantly changing, and the organisms, the communities, and the system have evolved to deal with this. An aside, I want to point out, most reef areas are not, in fact, high light. We think, oh, reefs are high light. They're actually, as I read that, I hear that reefs thrive in well-lit oceans. Well, taking the average attenuation of water, this is modeling light. This is the depth of the bottom. This is time of day, and the color is the intensity of photosynthetic active radiation of PAR. It's really only high in the first meter for 10 a.m. to about 3 p.m. On this particular no clouds. This is modeling, right? The rest of the reef in that high light. And guess where most of the reef is? Well, the reef flat will be down 2 meters, but in places that we like to go diving and snorkeling and like to see all the fish, that's all the blue. It's not high lights. I want to point that out. It's fun to point out. All right, more lecturing. I'm going fast because I've made too many slides. So I apologize, and you can ask me questions. Happy to answer. Light capture. So you get light to the sea floor. Congratulations, that's great. You made it through the lagoon. You made it through wherever. The things living there have to capture the light if they want to do anything with it, if they want to do photosynthesis and all the things that come after doing the photosynthesis. If there's going to be primary production, you got to catch the light. So here's a library that I have. Actually, I measured the library for reflectance, but this is 1 minus reflectance. This is absorptance to tell you how much light's being absorbed. And so all of these go from 0.6 to 1 for absorptance. 1 being perfect, it'd be a perfectly black hole if it absorbed 1, right? Nothing does. But sand here has a different scale from 0.2 to 1, just to point that out, because sand is brighter, so it has less absorptance. By far, the density of the spectra, so again, 400 to 700 nanometers, it's visible light, there's the violet to the red. Things are absorbing 90% or more of the light. They're absorbing a lot of the light, except sand. So reefs are absorbing a lot of light. That's good to know. The different benthic types, there's and those I have those for an old reason that dates back to my PhD that I never deviated from. Maybe I should. The benthic types have different shapes to them. That's because they have different pigments, which I'm sure you all know this. I'm sorry if it's redundant or boring to you. They have different pigments, and that lets them absorb different wavelengths a little bit better, a little bit worse than the next benthic type. And that can lead to ecological partitioning. That's sort of a well-known idea. Within each benthic type, there is a lot of variability. So there is no one brown coral spectrum. There's a lot. And there may be a mean, and there may be trends, but there's a lot of variability. All right, that's it about light. I want to move on a little bit to metabolism. So Don Kinsey asked the question, how can a community have a metabolic entity? So I have metabolism, you have metabolism, right? How does a community, how does this room have a metabolism? Have everyone in the room together? The answer is it doesn't. It's rates at which materials are imported, incorporated, converted, turned over, excreted, exported by complete community. Those words sound familiar because it was on my 3rd slide, right? So we're talking about the rates that things are happening among the community. We could put an oxygen sensor in the room and measure our collective respiration, right? I don't know what we would do with that, but we could do it. So reef metabolism is dominated by benthic autotrophic processes: production, respiration, calcification, and dissolution, right? This is, again, basic primary production and calcification. These are the these underpin everything. You don't have these, you don't have the reef. And so, important. Just a fun thing showing, it's just fun, got to show a video now and then, deploying the gradient flux mooring on Palau. I don't know if any of you know, any reef people know Bob Carpenter, and this is his former postdoc is now at KAUST, Chiara Pispia, deploying, there's flow sensors and oxygen sensors, and you measure the gradient of oxygen and the flux of water, and then you can from that calculate the production. And that was kind of cool. So that's one way, that's how we're measuring production now. Based on that flume experiment I showed you earlier, we were able to get daytime gross primary production, daytime respiration, and nighttime respiration. So full 24-hour respiration. Respiration, of course, does go 24 hours. Primary production is only in the day, right? And the lines are the averages, the dots are all the individual observations. So just to kind of show you the patterns. And for this community, respiration, so it's negative. So if I say it gets smaller, it means it's getting closer to zero. Respiration gets smaller from midnight towards sunrise. Gross primary production takes off with sunrise. Respiration lags and takes off a little bit afterwards. Gross primary production follows the sun mostly. Respiration builds up through the day and peaks later in the day and then tails off again. That's what I wanted to say there. These rates vary by reef zone, and that's not surprising, given that the reef zones have different depths on average and different water clarities, right, and different exposures to waves. So on the left-hand side is gross primary production and respiration on the y-axis, and the different symbols mean different bits of the reef. The highest rates generally are, as you see, this high activity area, these orange dots, the highest rates, and that's coral dominated, high activity meaning high flow environments. Then intermediate rates are in reef flats, which are the green diamonds. This is like Lucky Charms. Green diamonds, which are mixtures of coral, a lot of algae and a lot of sand. There are low rates for algal pavements, which are this guy right here and that guy right there. Lagoons are variable because some parts of lagoons will be entirely heterotrophic and some will be autotrophic. They're very mixed, very, very patchy. I want to point out this line here is the 1-to-1 line, right? And a lot of the dots fall close to 1-to-1. That tells us respiration about equals production. The net community production on reefs is typically around 0, right? Now this is production and calcification. This line is not the 1-to-1 line. Again, we see similar patterns in terms of the reef zones. The reason why we have this line here, it's the best fit line, is to show that calcification is roughly proportional to production. And that makes sense because the reef communities are getting their energy for calcification from production. You can have calcification at night, you can have it without production, but it's really strongly light mediated, with the production mediated. These are general trends. By the way, I want to point out where these data come from and where our knowledge of these trends. 1985, Kinsey's summary paper. There's some more recent that add on to this, but match the same patterns. Seasonality, summer, winter. Blue is production, orange is respiration. So production, respiration are kind of similar. And then winter, they're lower. So the rates in summer are about 2 times that of winter. Calcification, you have summer and winter. It's also different. The difference is 1.5 to 3.5 times summer to winter. We have these patterns that we're seeing, general patterns. Summer's higher, winter's lower. this zone has that, that zone has, you know, this. We have these patterns. And they're really useful. It helps us sort of conceptually understand how the reef is, how it's structured. But we don't have what's driving these things. Oh, I have some slides, not that you need them. Here's net ecosystem calcification, calcium carbonate production, dissolution. So so the net is this minus that. That's pretty easy. It's a budget. It's got to be bigger than 0 for the reef to persist. This is I'm treating you guys too much like students here. That here's some more pretty pictures of things. Corals, coralline algae, coralline algal pavement are responsible for the framework. Corals are like bricks. They die and these cement them, and then this makes the reef flat a lot of times. Bioerosion, mechanical erosion are responsible for breaking those down into sediments, which then sediment infilling is another process. These are all processes on the reef. Reefs are geobiological systems. The reef structure, this is equals structural and ecological coevolution over time. Things start growing, it builds habitat, lets more things grow, builds more habitat, builds more structure, more habitat, more things grow, more, it's co-evolving together. The structure and the biology and the ecology are going together at the same time. You don't have one without the other. You need them both. And all the interactions that are happening. This is just the reef framework building part of it that Glenn published in 1997. Nice cartoon, so I had to include it. But this is just about building the structure of the reef and arrows and feedbacks and things like that. So I already went through that. I'm rushing and I'm throwing a lot of stuff because I'm worried I'm going to run out of time, and I don't want to do that. Ecosystem function. So this I thought was a really great cartoon by Brandel et al. 2019. I really like this because it has these intrinsic drivers: calcification, primary production, secondary production, nutrient release. Right? I disagree with this whole side here, but you have bioerosion, herbivory, and predation. So you get up into the fishy things and all that, which are really cool. Now, I'm not talking about. I would just get in trouble, right? But ecosystem functioning is all of it, right? Is how the things are connected. It's the collection of all the processes working together. Again, to point out, the processes are not the ecosystem services, which is what we take out of them. Draw that clear. We harvest fish, but the processes are the herbivory and the predation and that kind of thing. The reef processes occur at high intensity and spatial density, but they're patchy. It can be. You can have patches, and it all depends on the environment. Every reef is different in that regard. Energy flow, calcification, nutrient cycling, and habitat provision are examples. We said, if we start losing reef structure, this was a word I couldn't think of earlier today, and it was really bugging me. Decouple. Oh, that's such a relief. Sorry. Decouple. What if we start messing with things? Can we decouple these? And how bad a problem would that be for the reef system? Functions may persist even if you do decouple things. Primary production will happen with algae or with coral. Calcification may be different. Knowledge gaps, as I see it. This is me. Production, respiration, calcification is a function of flow, nutrient fluxes, community structure, on and on and on. It's not reliant on one thing or another. It's a function of all of them. And we do not know that function, a mathematical function. Don't know. I told you, oh, high flow, low flow. That's a binary. What's the error bars on that? So this is what bugs me. It bugs me. Impact of acute versus chronic stress. These processes happen at different spatial and temporal scales. So reef accretion is hundreds to thousands of years. Calcification is minutes to days to a year kind of thing. It's different time scales. So acute and chronic conditions, stress, how is that going to impact these? What are thresholds for decoupling the functions? How much coral do you have to kill before it messes things up? What's the threshold? Is there a number? What are the quantitative connections? I like this, and this is beyond. This would be a great thing to work on, to understand the functions and the relation to the goods and services. How relating herbivory to the amount of fish you can harvest, things like that. And then how to scale. And I mentioned that earlier. How do you go from the observations that we can make by diving, which is great fun, but it's very limited. How do you scale it? And so I'm going to zip through, I don't know how many, as many as I can in the next 10 minutes, I guess I got. Scaling reef function, going for different scales. This is just a throwaway kind of slide. Here's the reef thing again, where reefs are. I changed the bottom. Quantitative in situ surveys are only tens to hundreds of square kilometers worldwide. That's just surveying for coral. That's not even doing the functional stuff. That's just the structure. So we've only surveyed the structure from 0.01 to 0.1% of the world's coral reefs, roughly. The only way to see that whole picture is remote sensing. It's the only way right now. Maybe in the future we have other ways. Question though is, how do you translate all the stuff I was just talking about to satellite imagery? Because that's a whole other thing, right? It was all reef science. I don't know if you've heard of the Allen Coral Atlas. They do habitat mapping. This is downloaded. This is Chook, the rim of Chook Atoll. They do habitat mapping. So that's the satellite picture that they used, and this is their map. And it's a beautiful map. Their classes are coral / algae, seagrass, microalgal mats, rock, rubble, and sand. From the perspective of understanding the reef system and its function and the interconnectedness, knowing that red is all coral or algae is of moderate use, modest use. It doesn't help me understand what's going on there. It's not a bad thing, but I think we need more. So, of course, that's what I've been working on a lot in my career. This is Apple Harbor, Guam. There's, I don't know if you've ever been to Glass Breakwater. There's the commercial port, the Navy port is down in there. This is all Navy base. This is where they store bombs, literally like stack bombs really high. It's fun to walk across. They say, don't go outside the yellow lines. Trust me, I won't. So Apple Harbor, there's a satellite picture of Apple Harbor. What we want is that, right? Where you have the benefit community map as colors of from all coral to all algae to all sand and things in between, right? That's what we want. And so this is what a lot of what my career has been working on. This is the problem of remote sensing. No problem. There's so many problems. Here's the satellite. Looks like a pair of pants with a rainbow coming out of it. There's atmosphere. There's glint. Then there's water. And what we want, if we're studying the reef, is this: the light reflecting off the bottom. That tells us about the bottom. Hopefully, it tells us a lot about the bottom, but we got to get that light first. A lot of smart people do this. Smart people do this. This is the issue, and then putting it together with that. Getting rid of the water. Now, Jonathan Lee had his wonderful HOPE model. There's his model for that. If we ignore the atmosphere, we ignore the sea surface, just the water and the sea floor. It's that. Whew. It's based on phytoplankton concentration and yellow matter and suspended sediments and a bottom brightness and the water depth and all these fun things and the angle and it's complicated. It's models upon models, and it works and it's good. I don't want to deal with all that. The thing is you make this model, you put it in code, and you tell the computer, here's my remote sensing measurements. There's a spectrum. I don't know what it is. Play with that until you match it. And when it's matched, I know what my PG, XB, and H are. Thank you very much. And now go do it. And the computer, come back and tell me when you're done. Too much for me. So I use a simplified version of that, which is this. And it's just backscatter, attenuation, bottom reflectance, and depth. There's 4 things instead of however many you have. Now, I've got data on attenuation in the water. It's great. I showed you some of it earlier in a different format. Wasn't specifically about attenuation. Again, wavelength, blue to red, attenuation in units of per meter. This is measured attenuation spectrum. It turns out, principal component analysis, fun dimension reduction technique. The first component describes almost 95% of the variability of this. So I have an idea. Let's do that. And this is if you model it based on the first principal component. It looks a lot like that. Hey, now the backscatter one is fun because it's a slope, right? It's really, really easy. The first component is 99.9% of the variance. Let's do that, please. And so there's the model version of the in situ measurements. So I can parameterize my model, not with phytoplankton and yellow, but just with one number for this and one number for this. Right? It scales it up and down. Now, the bottom type is harder. This is what I showed you before, but 1 minus that. This is reflectance now. And again, there's a lot of variability between classes and within classes. And getting that to be reduced to dimensions is a hard thing. And I've struggled a lot. I got a neat, I just like the animation. This is a self-organizing map, which is AI or deep learning clustering, essentially in 3 dimensions. Oh, it stopped. No. Oh, well. And it shows you how these things organize the spectra. And it doesn't organize very well. I mean, you got blobs here, here, and here, and all of the different things have different blobs in different places. It doesn't help very well. I'm not going to go on with that. This is hard. It's an area of active work. I told Brian earlier I have an idea how this works, so I won't give it away. So back to this to say that that was talking about the remote sensing model and how we build it and some things. I didn't want to bore you and delve into all that too much, but scaling up, we use that to, if we can make that model work, then we can have the thing match the spectrum to the model and say, oh, this is what's on the bottom. This is the coral or algae or whatever, the spectrum on the bottom. But what about the processes that I care so much about? Well, I stole this from the terrestrial field. Thank you, land people. Gross primary production is A par, absorbed photosynthetically available radiation times light use efficiency. It's a linear model, and it truly is linear, and I can tell you about it some other time. I literally was falling asleep, like I'm sure many of you are here, in a lecture when they started talking about it, and I went, wait, what? I could do that. And so here I am all these years later. We can expand that. It's par, which is the available light, times FA par, which is the fraction of absorbed par, times light use efficiency. If you want to break it up spectrally, which we might have to because we know red light goes away, there's different colors, different depths, we can say, move the efficiency over here. It's the integral of downwelling irradiance against wavelength times the absorptance integrated over visible range. That says how many photons are in the plants and what's the efficiency that the plant has at converting that to carbon, fixed carbon. We can rearrange that, which I do here. Now epsilon is over here. Light efficiency it's just normalized primary production. It's normalized by absorbed light. That's all it is. And I have all this stuff here that goes into, which I more or less copied from the paper. Put my own words, but basically, efficiency. If you're sick, you got a cold, are you working at peak efficiency? No. If you're a plant, if you're a tree, and you don't got enough water, are you working at peak efficiency? No. Right? Efficiency basically responds to the conditions. Like, how's the thing doing? And it turns out, in land plants anyway, that a lot of different land plants, you go, they're different, have convergent efficiencies. So it ends up being really, really, really useful. And in fact, there's a standard NASA product from going back to the beginning of MODIS, if not further, that uses this model without the integral thing. It's just all based on par to map primary production on land surfaces. This is the model they use. And the cool thing for me and for them, you can also, I know there's a software that you can buy called Envy. I don't know if you can still buy Envy, right? It's built in. It's off the shelf. There are tools. Oh, let me load my spectrum. You click it, and it will give you epsilon based on the spectrum, because people have done that research to relate the reflectance, which is 1 minus A, to that. So people are doing remote sensing on land and going, here's my spectrum, and getting an efficiency out of that. That would be way cool to do on reefs, because we know that this it depends, it's related to optics, but it's also related to the conditions of the thing. And so if we can do optics to condition, oh my God, we don't need to model GPP if we don't care about the absolute amount of carbon. We just want to know the condition if we're worried about reefs. So I've done some work on efficiency. I've measured some, don't worry about this, but this is the data that goes into calculating the efficiencies, and it partitions coral, algae, and sand. And they overlap, and that's with very little data. There's a lot more work to be done there because I don't know what conditions they were all in. We could play with that for years and refine it. But the idea then is, hey, here's my map that I showed you earlier of coral, algae, and sand. Let's apply the efficiencies and estimate primary production. Right? That's the idea of scaling up metabolism. I'm going to get into a little bit of just where we were, where we're going, and again, I know I got to stop. 1978 reef map from a satellite. Can you read those? There's 33333313330. That's awesome. That's what the reef actually is. It's Apo Reef in the Philippines. Remote sensing has come a long way, right? At least displaying the images. They didn't have the ability to display it or print it out anyway. So there's some questions about scaling. When we go diving, I like to see the coral. I like to see the fish. That's at like 2 meters, like 2 meters tall, give or take actually. So there's a reef. Great. That's the reef, 30 meter pixels that a satellite might give us. How? Do we need this? Is this okay? What about bigger? Is bigger okay? What's going on in the mixture? What does that do to our understanding of the structure and of the function within that pixel? It's as big as this room. Like, I'll swim along from here to there and back and forth, take me an hour to look at every little detail and the little shrimps and the octopuses and things like that. That's awesome. What about the processes I'm talking about? Right? This is close up. You see the scale here is 500 meters. Here the scale is 5 kilometers. When you look at the reef at that scale, it doesn't look any different. So I say, for what I'm interested in anyway, I say 30 meters is fine. 60 meters. Let's see if 1 kilometer works. But there are issues with that, right? So another thing about remote sensing of reefs is that clouds. Oh my God, clouds. This is all the Landsat scenes for Palau for 2018. And so there's no one picture you can use. You're not going to get day-to-day changes. You're going to get long-term. And you got to be okay with that. What does that mean for your science? What does that mean for understanding the reef processes? Got to be considered, because you're not going to get daily clear pictures places like Palau. Trust me, you're not. I didn't know. All right, this is I said view-dependent atmospheric correction is a must. Atmospheric correction is an issue in ocean color remote sensing, and when we did for the coral mission, they assumed a nadir view, but the sensor had a 36-degree swath field of view. So the plane going north on 9 a.m. on Guam, the sun is over there. And so we get a lot of glint on this side and a lot of atmospheric backscatter on this side. After we remove the glint, so this line here, this is a just one wavelength. This is the pixel values. After we remove the glint, those are the pixel values. This is incomplete atmospheric correction. We have atmosphere left in the picture, and that gets in the way. It causes problems. And so I say we must have better atmospheric correction. And then I say, well, we're doing. This is an idea. It's unpublished. Steal it. Fine. Here's not the same picture, but here's a pixel in blue. Here's a pixel in red. Those are the spectra now, right? From 400 to 800 nanometers. The red is on the glint side. The blue is on the high atmosphere scatter side. And you can see they're different. And if I ran those through by optimizing my retrieval, I would get different answers. They're both deep water. It's the same water type, same water stuff. But they're different because of the atmosphere and the sea surface. And people work hard at fixing that. We could just take the second derivative, and they're pretty much the same now. And so do the retrieval. Brian, I'm giving it away to you right now. Do the retrieval in second derivative space. I talked about scaling. I don't know why I have this again. It's a pretty picture. Here's a nice reef in the Great Barrier Reef. It's a really pretty orthomosaic. That's about 8 meters on a side, right? The pixel for the coral mission that we did would look like that. So that goes back into the question of, I'm repeating myself now, and I'm doing it again. I apologize for that redundancy. Same deal, right? And then, I didn't talk about was depth. So this is reef from the previous picture, and it's not just what's there, the biology, but the depth. So here's a little reef down here. That's what it looks like. Are we going to get like, the processes is going to be messed up? Well, we have to understand the scales of the processes based on the scales of our observation. That's what I'm trying to get across. All that sort of ties together and comes out of the coral mission, the Coral Reef Airborne Laboratory. Here you go. We flew a plane over Hawaii, Great Barrier Reef, Palau Islands. In Florida, we did the Keys as well. We didn't do any field work there. The plane was coming back from Palau, and it was based in Maine, of all places. And coming back, we look at the weather. We had money left over to fly. Expensive airplane. We had money left over, and the weather was good. And so they flew all the Keys all the way up from the Tortugas to Fort Lauderdale over a couple of days. So we have that, no field validation for it though. And the problem is that the reefs in Florida, because of all the damn gorgonians, look different than the reefs. And so I'm keeping those aside. If someone wants to work on that with me, I'm happy to do that. But the idea is, here is the Torres Strait. Australia's here, New Guinea's here, 12 flight lines. We map coral, algae, and sand like that. And then what I'm working on for the reef science is you decimate that to 1 kilometer grids. So I figure a reef scale is about 1 kilometer. And so we can look at that coral distribution in relation to ocean acidification, thermal stress, wave stress, all these fun things, one by one, or we can throw them all together into a multivariate model, like I did. This was a bag ensemble of regression trees, I think it was, and go, can we predict coral cover based on these combined? And we can do that to about 64%, explain 64% of the variability, which means that there's at least 36. So putting aside problems with all the processing and the imaging and the errors and all that, if that's correct, there's still 36% of variability in coral cover that's not explained by the environment. And thank you.